Hello and welcome back to our series on plant hormones. In our last video, we covered the basics of gibberellins. Today, we are diving deep into the fascinating world of how plants actually make and control these powerful growth regulators. Where does this all happen? It is like a tiny assembly line spread across three different parts of the cell: the plastids, the endoplasmic reticulum, and finally the cytosol. This compartmentalization is super important because it allows the plant to tightly control which gibberellins become active. Let's start our journey in the plastid. The starting block for every gibberellin is a molecule called GPP or geranyl diphosphate. This molecule is a common player in plant metabolism. Through two quick steps, GPP is transformed into a molecule called ent-kaurene. First, an enzyme called CPS turns GPP into ent-copallyl diphosphate. Then another enzyme, KS, converts that into ent-kaurene. Think of ent-kaurene as the bridge molecule from which all other gibberellins will be built. This part of the process is very similar to how plants make terpenes, which you might know as the compounds that give pine trees their distinct smell. Next, ent-kaurene moves out of the plastid and heads to the endoplasmic reticulum or ER. The ER is like the cell's workshop. Here, ent-kaurene undergoes a series of oxidations, first by an enzyme called KO and then by an enzyme named KAO. These reactions are handled by special proteins that sit in the ER membrane. The final product of this stage is a molecule called GA-12. GA-12 is a huge deal because it's the universal precursor for all other gibberellins. It is the gateway from which the plant can decide to make a whole variety of different GAs, both active and inactive. Now GA-12 travels to its final destination, the cytosol, which is the main fluid-filled space of the cell. Here things get interesting because the pathway splits into two main branches. The first is the 13-hydroxylotidone pathway. In this route, GA-12 is converted through several steps into GA-1, which is the primary bioactive gibberellin in many leafy plants. Two key enzymes, GA-20 oxidase and GA-3 oxidase, are the stars of this show. The second branch is the non-13 hydroxylation pathway. This route takes GA-12 and, through a different series of steps, creates GA-4. GA-4 is another major bioactive gibberellin, especially important in plants like Arabidopsis and many grasses. So, depending on the plant and its needs, it can favor one pathway over the other to produce the right active gibberellin for the job. But what happens when the plant has enough active gibberellin? It can't just let it build up, or it would grow uncontrollably tall and spindly. This is where regulation comes in. The plant has several clever ways to keep GA levels in check. The most direct method is deactivation. An enzyme called GA-2 oxidase takes active GAs like GA-1 and GA-4 and converts them into inactive forms like GA-8 and GA-34. These inactive versions can no longer bind to the GA receptor, effectively turning them off. Plants also use other tricks, like adding a sugar molecule or a methyl group to active GAs, which also neutralizes them. Beyond direct deactivation, plants use a classic feedback loop to maintain balance. It's simple but effective. When active gibberellin levels get too high, the plant shuts down the genes that make the key synthesis enzymes GA-20 oxidase and GA-3 oxidase. This slows down production. Conversely, if GA levels drop too low, the plant ramps up the expression of these genes to produce more. This elegant system ensures the plant always has just the right amount of gibberellin for healthy growth. Finally, there's genetic control. In the shoot apical meristem, the very tip of the growing shoot, special genes called KNOXI genes actively suppress gibberellin synthesis. This is crucial for keeping the meristem cells in an undifferentiated, stem cell-like state, preventing them from elongating too early and ensuring the plant can form new leaves and flowers properly. And that's a wrap on how plants produce and meticulously regulate gibberellins. From a simple starting molecule to a complex web of synthesis, deactivation, and feedback, it's a beautiful example of biochemical precision. I hope this gives you a new appreciation for the hidden chemical factories running inside every plant around you. If you found this interesting, don't forget to like this video and subscribe for more deep dives into the world of plant biology. Thanks for watching and see you next time.